Good morning, everyone. Welcome to the October 3rd Thursday, not October, September 3rd Thursday. I'm ahead of myself because I think Crush is ahead of itself. Full disclosure, I'm a little sticky here. So yeah, we're going full bore pretty much on the Crush pad. So I hope everybody else is too. So of course, this recording will be here for your sticky colleagues when they get off the Crush pad. So today we have a presentation by Dr. Paul Rivarez, who goes by Paul, and he's a new professor at UBC in Vancouver, where he leads the Phytopathology and Virology laboratory. He specializes in plant pathology, virology, metagenomics, and bioinformatics. And he earned his PhD from the National Institute of Biology in Slovenia as a prestigious Marie Sklodowska-Curie Fellow. How was that?
Yeah.
And conducted postdoctoral research at North Carolina State University. I'm gonna hand things over to him. He's got a couple of really interesting topics that I think we'll be hearing more about here in the wine industry. So welcome, Paul, and take it away.
Yeah. Thank you, Kathy, and thank you, Kate, for organizing this. And thank you to the rest of the BC Wine Grape Council for inviting me over. And I'm happy to see some colleagues in over Zoom joining us. I hope we will have like some fruitful discussion towards the end of my presentation. And the goal for me is, to just have like a little bit of fruitful discussion on how we can put together a concept, a project that will help with high-throughput virus diagnostics in grapes, especially for on-site approach, also laboratory approach, but more on the on-site approach. There you go. So for my presentation, I will give you a little bit of background about virology in general, and especially about Solanum nigrum Ilarvirus 1. And then give you a little bit of the research aims and the methods that we used in order to study and dig deeper into the properties of SNIV1. I will also discuss some results and summary and some future plans. Of course, acknowledgement towards the end, and question and answer. So just a little bit of introduction on how high-throughput sequencing impacts virology. So virology as a field of study is very young, I would say. It started with the discovery of tobacco mosaic virus around 12 decades ago. So it's in that sense, it's very, very new compared to other fields of research or study. But if we look at records from millennia ago, we can see that there are records of this disease symptoms described in Eupatorium yellow vein disease from Japan, some 1200 years ago. So the field has been existing in terms of like descriptive symptom description, but it has gained track with the discovery of the tobacco virus, its mosaic virus itself. Fast forward 12 decades. Now we have the idea of virus diversity organized in 15 ranks of megataxonomy spanning 6 different realms. Right now, the International Committee on the Taxonomy of Viruses recognizes more than 16,000 species of viruses officially. But then the virus sphere is very vast. So the whole collection of viruses, viroid-like agents, and anything in between, it's really vast. And from estimates, there are around 80 million species of viruses, and we are just uncovering a few 100,000 of them. So in recent years, in silico or data mining exploration of sequencing data led to the discovery of a few 100,000. And then this is just my estimate. Now, cumulatively, we have around 300,000 viruses species identified to date. And of course, vastly are uncharacterized, which is approximately 5%. That was the figure before that I showed you, which is 16,000 species. So there's still a lot of work to do in terms of just characterizing the full set of virus species that can be on Earth. Now, focusing on one virus out of the thousands of viruses possibly there or millions. So we focused on Solanum nigrum Ilarvirus 1 or SNIV1. It is a new Ilarvirus associated with Solanum nigrum plants and tomatoes from France. So that was the first time that the virus was detected in 2020. What's interesting is SNIV1 was concurrently detected in various virome studies involving peas and other weed plants from Germany, Plasmopara viticola infected grapevines from Italy, also from 2020, Erythrophinacator infected grapevines from Spain, also from 2020, and wild Solanum species from South Africa, and then in Slovenia as well, which was reported 2022 and 2023. So from my title, the virus has several names. It's 4 of us names in the literature, and so it's quite confusing in a sense. So this virus has 5 names, but it's really just one virus, just because the high level of or high magnitude of high throughput sequencing happening in that particular period led to the concurrent detection of this virus, and of course different namings and all that. But in a sense, it's Solanum nigrum Ilarvirus 1. So the detections were mostly made in Europe and one in South Africa. So we dig deeper into the virus and characterize it a little bit. We were able to isolate the virus particle, but Solanum nigrum Ilarvirus 1 being an Ilarvirus or under the family Bromoviridae, it's very tricky to isolate intact virions or virus particles. So we were lucky enough to see physical evidence of the existence of the virus, which is from literature we know it's typically 25 to 27 nanometer in diameter. So as I mentioned, Ilarvirus is under the family Bromoviridae. It's closely related to cucumber mosaic virus, alfalfa mosaic virus, and so on. And this is the clade to which it belongs with the 4 different names. And interestingly, as I mentioned, multiple times the virus was detected in grapevines. So we were curious about that and how, why did it happen? Like, why is it detected in both grapevines and also weed species, Solanaceae species in particular? So we dig deeper into that, which I will discuss later on. So just a brief background. So the family Bromoviridae has 3 segments in its genome. It's RNA based and composed of 5 proteins. So we went back to the field of grapevines in the Indrae complex in Bordeaux in France. So we tested grapevines. We also tested Solanum villosum, which is another Solanaceae species, and Solanum nigrum, of course, where the virus was first detected. So in terms of co-location, here there is a soil mound where Solanum villosum plants are. This is another section where another Solanum villosum plant is, and there's also, of course, there's the vast grapevine fields research fields in the right complex. So they are pretty much very close to each other. And yeah, so from initial detection, we were able to really detect the presence of the virus in both the weeds and the grapevines. But that's from field samples. So we are curious then, like, to ask the question whether the virus is really infective. When we tried to inoculate the virus to the host plants. So in essence, we were able to formulate certain questions around the virus. For one, where else could it be in the global data set of sequences so far? So luckily, during that time, there was this tool called Palm ID search, wherein you can input a sequence of the virus, and then it will show you where that virus is in a sequencing data set across the globe. So we were able to know some locations of the virus where it was not reported before. So that's the first question. We mined nucleotide databases. We did additional sequencing of plant samples. And then upon assembling the genomes, then we asked the level of diversity among global isolates. So we did diversity, phylogenetic analysis, and phylodynamic analysis. And then third, if the virus is really infecting those plants, where is it localized and how it is transmitted? So we did a little bit of studies about transmission and also localization within the host plant. So we did some RT-PCR tests on different parts from roots to pollen. And also we did mechanical, seed, and graft transmission assays. So that's the whole set of questions that we wanted to answer, all leading to the ultimately like making sense of what really is SNIV1. Is it affecting grapevines? Is it just a virus that's lingering in the weeds? So we were able to have a sense of that in the end. Now for the specific results, so we conducted 4 different virome surveys, additional ones that we did before. So we employed 3 different techniques. Ribosomal RNA depleted total RNA for one. We also did double-stranded RNA sequencing, and we also did virion-associated nucleic acid sequencing, combination of a nanopore and Illumina platform from France, with samples from France and Belgium. So with this effort, we were able to add 7 SNIV1 isolates with full or partial genomes. So we got good coverage with some of the samples. Some have low coverage, but still full genomes were recovered. So when we used the Palm ID search, we were able to detect SNIV1 in 80 SRA datasets. Within those datasets, 21 SNIV1 genomes were assembled, bringing the current total to 34. So we were able to add more genomes to the database, SNIV1 genomes to the database. And then we were able to detect the virus across 6 out of the 6 habitable continents, or 11 countries. So we were able to expand then our knowledge on the distribution of SNIV1 just by doing database mining. 15 out of the 25 datasets that has SNIV1 are non-intentional. So meaning those studies that did sequencing before, they have different questions in mind, different research topics, but it just so happened that SNIV1 was also sequenced in their efforts. So that was unintentional in a sense. So with the SRA datasets or with the global datasets, we were able to sequence or assemble the genome of the virus with very high coverage, like ranging from a few thousand to like 77,000 times coverage. So we can say that the virus is really there in the sequencing dataset or in the sample. One interesting thing is the presence of the virus in Lithosperma erythrorhizon, which is a medicinal plant. And then you're also able to detect the virus in honeybee intestine datasets, which leads us to some hypothesis that maybe the virus is pollen transmitted. So we dig deeper into that as well. So yeah, I'm showing here the overall global distribution of the virus after our sequencing efforts and the data mining efforts. So you can see most of the viruses, most of the SNAV1 isolates that we were able to assemble were from Europe. Some were from the US, and then some are from South America, and then one is from South Africa, one is from Japan, and one is from China. So five out of the 6 habitable continents except Australia. And then what I'm trying to show here too is, most of the hosts or, the sample data sets were plant samples, and some are from fungi and some from Corista, which I already said before, so Erisipe and Plasmopara. But interestingly, as I mentioned, we were able to detect the virus in honeybee sequencing data sets. Also, interestingly and quite confusingly, we detected the SNIV1 in a human blood sample. So that leads us to more questions. Is the virus really a plant virus, or is just something like a contaminant in a sequencing data set? But it having been seen this, like our hypothesis of the virus being pollen transmitted got stronger, because if the virus is pollen transmitted, then it could be in the environment, and it so happened to contaminate some samples from other sequencing efforts. For example, this one, the blood sample is from Italy. And so far, we were able to prove that 3 isolates were plant pathogenic. I will show you the results later. 8 isolates were from sequencing of non-plant samples. For example, like I mentioned, fungal and oomyid samples. So in terms of the evolution of the virus, we using phytodynamic analysis, we were able to trace the origin of the virus from Europe, from the European continent, as the possible origin. And interestingly, from this phytodynamic analysis, we were able to estimate that the virus has recently emerged. It has just recently emerged from 125 years ago. So in an evolutionary sense, that simultaneous detection of the virus makes a lot of sense if it has emerged recently in the evolutionary timeline. So of course, if more samples, more virus genomes were sequenced, we can make more sense out of its distribution and evolution and possible origin. But now our analysis lead to its origin in the European continent. Now, in terms of infection in plants, as you can see here, in A, we were able to show its infectivity. Actually, in all of this, these are all Solanaceae plants. So we have Nicotianas, we have Solanum nigrum in the picture here, which are were all positive in RT-PCR when we inoculated them either through mechanical inoculation, approach grape inoculation, and chip bud inoculation. So the virus is infecting Solanaceae, and that's for sure. And so consequently, in the same time, we were also testing the infectivity of the virus in grapevines through graft inoculation. But unfortunately, or actually fortunately in a sense, we were able to prove that the virus is not infecting grapevines, which is a good thing. But we know that the virus is present in grapevines. So there's still some risk associated with it. But yeah, we're sure that it infects Solanaceae species. So just here are more details about our in infectivity assays. So we're able to detect or to prove its infectivity in Nicotiana, Bentamiana, occidentalis, and some Solanum species, specifically nigrum and villosum. So overall, we were able to answer our questions that I presented to you before, with more questions being asked or being formulated in the end. So we were able to detect SNIV1 in a range of countries in a very diverse set of plant and non-plant sequencing data sets. So now there are 30, after the study, there were from around 5 genomes of the virus, we were able to put more in the database, and now there are 34 genomes of the virus in the database. In terms of diversity, we were able to show in our phylogenetic analysis that the diversity is low, and the phylogenetic resolution is also, it can be improved with more isolates sequenced. So, and yes, for our third question, we were able to show its infectivity in Solanaceae species, but fortunately, in grapevines, right now, there's no evidence of being it infective in grapevines. But yes, as I mentioned, we know that it is present in grapevines. So this study was published in Phytopathology. So if you want to dig deeper into it, the paper is open access, and you can read the full thing. And also, all our data sets are uploaded, and so on. So yeah, and how about some plans, which I'm excited to talk about with you all today. So in my lab here at UBC, so I started roughly like well, now close to 6 months ago in April. So my lab will be focusing on the phyloecology and agrobaromics of viruses. So what is it about? So we are interested at the presence and dynamics of virus populations in the agroecological interface. So the exchange of viruses and the coexistence of viruses with weeds and also with crops, and the possible spillovers and the evolution, the evolutionary dynamics that come with it. So we are interested with both viromes and microbiomes in a sense. And having been said that, the only way to study them deeply is to develop high-throughput diagnostics. And so that's the goal of the lab to start with, to ramp up diagnostics, especially high-throughput sequencing, and also not just in the lab, but also sequencing on site, which nanopore sequencing will be really useful for. So in a sense, we are concerned about the hosts, which are the crops. Some of the viruses also replicate in insects or arthropod vectors. And so we are also, of course, interested about that. Of course, the environment is one aspect that maybe in the future we will touch on, because we also have experience in viruses that are present in the irrigation water or in the soil. And of course, the sequence mining that I mentioned to you a while ago, we are wanting to continue on that. And so in the lab, we expect some routine sequencing, data mining, and characterization. And of course, all this will not be useful if we don't have close collaboration with grower associations and councils, like the Wine Grape Council, for example, and other organizations across BC and Canada. And so we wanted to work closely with this organization, so that research is grounded on risk assessment and also policy development. So we expect a lot of active coordination. So our first aim is we wanted to analyze the relationship between host, the virus, and their coevolution to better predict potential host range, expansion, and spread or emergence. And with vast sequencing data and some bioinformatics analysis, we can all make sense of that. Second aim is for us to characterize the pathobiological properties, infectivity, co-location, location in the plant, transmission modes, so that we can assess the risk, okay, of the virus that we detect, and for us to be prepared in possible outbreaks. Third is to identify the diversity of end reservoirs of viruses in crops, wild plants, in the environment, and like I mentioned, in the interface of the agroecosystem. Of course, again, using nanopore sequencing, and on the side, Illumina sequencing as well, but we wanted to really emphasize on nanopore sequencing. And then the fourth aim is to proactively monitor the global distribution and spread, host range expansion, and identify new virus species or ecological niches using data mining. And in a sense, that's very doable nowadays, because high computing power is very accessible here in UBC. We can also set up in the lab some computing facilities, bioinformatics, and so on. So yeah, we expect this to be really active. Of course, to start with, because setting up a molecular biology lab takes a lot of capital, so we are expecting to start with actually some bioinformatics. And so in a sense, we want to really scan and scan the needs of the agricultural industry in BC, what is really needed, and what are the gaps in terms of diagnostics, and just in a sense, in general, like pathogen ecology, evolution, and epidemiology, the triple E, so to say. So right now we're connecting with the Grains Association across BC, floriculture industry as well, fruits, especially blueberries, and of course the grape and wine industry, which are all valuable industries in BC. So as I mentioned, we wanted to see the virome dynamics and how the viromes changes, also in association with other diseases and the changes in climate. So that is our goal. We wanted to set up some experiments in the UBC farm and extrapolate the data in a province-wide sense. So these are just some possible sources of funding that we are thinking about, including, of course, Genome BC, NSERC, and internal funding from UBC. So some relevant experience that I've got. This is me in North Carolina University, where I developed this workflow for efficient nanopore sequencing of tomato spotted wilt virus. This protocol is very, very useful, and I wanted to have it applied, in a sense, in one of the crops here in BC. And in the Philippines, at the Philippine Genome Center, I kind of transferred those skills and went back to my home country, which is the Philippines, and trained students and researchers on nanopore sequencing, which also I will be really excited to do here also in BC. And yeah, overall results that we expect, of course, the comprehensive understanding of virome dynamics associated with major agri-produce in UBC and, of course, the BC province in general. Democratizing of sequencing technologies. So we don't want our sequencing technologies to stay in the lab. That's why I wanted really to start using nanopore sequencing, because you can bring it to the field, and farmers and other agrologists, plant pathologists can also experience it themselves. And of course, ultimately, plant pandemic preparedness, getting one step ahead of virus evolution and pathogen evolution, in a sense too. So in a sense, the nanopore sequencing platform can really be inside a small contraption, which you can bring in a field, just like what I'm showing here. So the full complex sequencing that comes with, for example, larger platforms such as Illumina, can be condensed in a lab-in-a-box concept. So nanopore sequencing is an example of like a portable platform that you can bring in the field for sequencing. And it has benefits of being really rapid and very quick in giving results. And that's about it. And thank you so much for listening. And I'll be happy to answer questions.
Thanks, Paul. When you talked about the lab, I've been talking to people about a handheld device. It's a little bit bigger than that, the lab in the box.
Yeah. I mean, the device is very small. It's about the size of a smartphone. But all the peripherals that's needed to make it function, of course, can be contracted.
Yeah. So. Jose, do you think this would be useful for your work?
Hi, Kathy. Hi. I actually met Paul, and we have had already conversations. And it was very kind of him to reach out to me when he just landed at UBC, and so I really appreciate that. And we were discussing actually the project, and he was pretending. I think that's the ultimate goal, and we know, I mean, we have been hearing that from growers. Like a platform that can be used on site. I mean, ultimately, something that, as you said, a small device like a phone that you can, we have things like you take a picture and they can tell you, maybe the symptoms, what is associated with, or ideally take a sample of a leaf and process in the field right away after few seconds or few minutes, tell you is leaf rot, is red blood. Those are ideally the end goals we are having. So I think it's technology is getting there, and it's moving faster than my brain. So I'm glad that young people like Paul bring all these perspectives, because it's even very hard to catch up for myself. But I think it's useful. I think it will be there. I mean, there are other issues regarding practicality, cost, and things like that they still have to be, because we can design the best detection system, but if it's going to cost us $1,000 a sample, that's is the same as a fungicide or a biological control. We can put out there a fantastic control, but also we have to look into that. But no, I think it has a lot of merit, and I think is a way to go to start with the technology we have right now, and it's evolving every day, looking for these solutions. I think it's very good. So I have a couple of questions from Paul. It's the first time I see the presentation. I was chatting with him before, but I'm very curious about the detection of this virus in grapes, as you mentioned, because you said, well, we can detect it on grapes. It doesn't look like it's pathogenic or it's causing any symptoms on grapes. But you mentioned at the beginning that most of the detections were either in powdery mildew or downy mildew, infected plants. So I guess it's probably a direct association of the fungus with the virus, and we know there is sort of mycoviruses that they will transmit. So that, but my question is, can you detect in the plant, inside the plant, without those infections happening? Is the fungus flowing limited? We have over 100 viruses identified on grapes. Not all are pathogenic, so I don't know if this can fall in one of these many viruses we have on grapes that I mean, yeah.
Actually, thank you for asking that. I actually failed to mention, like, on our detection or like the diagnostic efforts in grapevines, we always make sure that we don't include surface tissues. So we exclude epidermis for one in the bark. We scrape the inner layer of the stem. So we don't do diagnostics on the outer parts, so that we make sure that contamination from the environment is excluded. And that having been said, that we are always sure that if it's negative, it's really negative. It's not in the plant or replicating in the plant. Yeah.
Okay. No, it's very interesting. Yeah, it's I think every year we find, I don't know how many new viruses viruses on grapes. It's a crop that it has quite a but I mean also to be not all of them are causing problems or so no, this has very interesting presentation, not very very good.
Yeah, and in a sense, I could probably expand a little bit. So like you said, it was detected in powdery and downy mildew pathogens. But when we isolated those pathogens and we picked colonies and we tested for SNIV1, we didn't detect SNIV1. And then I mentioned it very briefly in my presentation, but yes, we were able to detect SNIV1 in pollens. So it is characteristic of Ilarviruses and some Bromoviridae species to be transmitted through pollen. There's been studies from decades ago that proved that already. So yes, and we just like proved it another time around that it is really pollen transmitted.
I'm wondering if, so you say that there a large number are not pathogenic, but does that mean that they really have no effect? Do you think that there's a possibility they might have a positive effect?
Yes. In a sense, survivability of the virus is enhanced if, for example, this pollen transmitted virus stays on a grapevine and survives in a grapevine because the grapevine has a longer it stays in the field. It's not like annuals that you harvest and you replant again. So in a sense, survivability of the virus is enhanced, not directly in terms of its pathogenicity or in a sense of virulence too, but in terms of long-term survivability and environment that helps. Also, I mean, I could expand a little bit, but when a virus infects different host plants, their evolution in a long run is assured, and their survivability and fitness is also assured in the environment. So being effective in many species helps in an evolutionary sense. But in terms of effects on like the health of the plant and all that, most viruses, yes, you're correct, don't really affect the health of the plant. It's just some viruses they choose to be infective in a sense, like in their evolution it is their trajectory. So they to be evolving as a pathogen, that's their trajectory. But most of the time, some other viruses just stay in a neutral relationship or a coexistence with its host.
But to be fair, also, and maybe Paul can add into that, we have a system with grapevines with so many viruses, viroids, that we consider not pathogenic. As we have seen and observed during all our knowledge we got during the pandemic and the COVID, viruses can mutate very, very quick, very fast. And that's something that, we don't know. It's kind of a, ticking bomb where is any of these viruses able to mutate to a point that needs to adapt and cause, or also we know by fact that there are some viruses that by themselves they will not cause any harm, but if there is a mix or combination of some other viruses, for example, I'm talking about rupestris stimpeting, by cells, it's not really harming, but if we have rupestris stimpeting with GBA, GBB, that's where we are still seeing more issues. So that's also where we have a system or a plan with many viruses that they don't cause harm, but is in the evolutionary aspect that Paul mentioned, we don't know what can happen, and that's what we have been finding sometimes.
I totally agree. We don't know what could happen. It's really unpredictable. But in a sense, what's good with what we did with SNIV1, we were like a few steps ahead. The virus we estimated recently emerged 125 years ago, which is in an evolutionary sense very, very recent. So we are like we were able to monitor the virus before it caused some problems, and that's actually true. because after we published the study, like I think it was 1 year, 2 years later, the virus was found infective and causing problems in peppers in Greece. And so so it's just, well, maybe we're a little bit still, in a sense, like in our researches, we're still a bit slow, but the virus can, like, if it evolves to be pathogenic, then it can just quickly, like, cause outbreaks. And that's what we wanted to be, like, a few steps ahead of that, like we monitor the virus before it causes problems.
Hans, I was going to ask if you had any questions.
Well, thank you for that. Yeah, question on the nanopore sequencing. I mean, in layman's terms, how is that different from the Illumina platform that has been around for quite a while? And also, the application in the field, is this actually field ready now, or is this a future prospect? And what would it take? How long would it take? Could a sample actually be processed in the field?
Right. Yeah, Hans, thank you so much for those interesting questions. Nice to meet you, by the way. In a sense, nanopore sequencing is a little bit different with Illumina, I would say, in terms of it sequences long reads, whereas Illumina is quite fixed on short reads. Can be 200 base pairs, 250 base pairs. But nanopore sequencing can sequence longer than that, like a few thousands or even millions of bases in some cases. So and it can sequence the native form of the genome, so meaning no modification aside from being mechanically separated or dissected in a sense. But yeah, it can sequence the native form of the genome of the virus or of pathogen or any other organism. So short reads versus long reads, that's basically the difference between Illumina and nanopore. But in terms of you asked about it being applied to the field situation, yes, it has been applied in a lot of cases already. It has been if I'm not mistaken, like in the past 5 to 10 years, it's been actively applied in the field. For one good example is Puccinia graminis whole genome sequencing in the UK, where they have nanopore sequencing on field, on site. So it's been used to monitor pathogen evolution of Puccinia, so wheat rust. So and also for non-plant diseases, so in humans, nanopore sequencing is very useful for I think they did it in Brazil with Japanese encephalitis virus and other like Zika viruses as well. So this platform has been deployed quite extensively already in the field.
Yeah, thanks. And in terms of the affordability, I mean, is this at some point going to be affordable for larger industrial entities in agriculture?
Right. So if we're talking about on-site diagnostics, right? So the capital is really low. Well, to establish the device, it's right now in Canadian dollars, it's a little bit over $4,000 just the device. But of course, if you want to deploy it in the field, you need to have the reagents and all that, which is like a few grand. So if you have, like, for example, like 6 or 8 grand as capital, then that's already a good start to deploy the device in the field.
And the software, I mean, now you need access to a fairly extensive database, right?
Yeah, that's one good thing. With Nanopore, sorry, just disclaimer though, I'm not associated with Nanopore in a sense. I'm just a researcher using Nanopore. I'm not promoting Nanopore, but yeah. So Nanopore, they have open source software which you can access easily over the internet. So if you have data or sequencing running already, you can analyze your data real time. So at the same time, like you're sequencing. So, you know, at like 30 minutes to an hour, you already have the idea of what are the pathogens in your sample. Yeah. And it's free.
Thanks, very interesting.
Yeah.
Any other questions? Kate says we don't have to go the 4 hour. She's given us permission to end early. So if there aren't any other questions, we will thank Paul and let you know that we're excited about this. I think everyone's minds are going, thinking about various applications. And so welcome to the research community.
Yeah.
And yeah, I guess I'll go back on the crush line and get sticky again. And we'll see everybody in October. I think that topic is TBA, right, Kate?
No, Dr. Mehdi Sharifi is going to be giving a presentation, a bit of an update on his travels to the GSCO conference and the things that he learned there about European vineyards and how we're comparing here in BC.
Okay, look forward to that. That's October 17th, correct?
I think it's the 16th.
I was so close.
You're close, 16th.
Thanks everyone for coming. The recording will be on our website. So, you know, because I think it might be difficult to explain to other people. So they can have a look at that. And thanks again, Paul. We'll see you again sometime, I'm sure.
Yeah, of course. Thank you so much again for inviting me to this.
Our pleasure.
Thank you.
Okay, bye everybody. Thanks.
Happy harvesting. Bye-bye.